Welcome to the Theoretical Neuroscience Podcast. I am the host, Yiota. If you study the brain through a microscope, the most striking feature is the beautiful patterns of branches that protrude from the cell body of neurons, what we today call dendrites. Santiago Ramon y Cajal, one of the founders of neuroscience as a scientific field, poetically referred to these beautiful neuronal structures as butterflies of the soul. However, the functional role of dendrites remained elusive for a long time and is still not completely sorted out. A major breakthrough came in the 1960s when Wilfred Rall demonstrated through biophysical modeling and the use of the Cable equation that dendrites can play a crucial role in determining electrical properties of neurons and how they integrate synaptic inputs. This work also paved the way for what is now known as multi-compartmental modeling of neurons, and software tools such as Neuron, described in podcast number 15 with Michael Hines, has been developed. With such tools, electric properties of neurons with complicated dendritic structures can be straightforwardly modeled. Today's guest, Yiota Poirazi, working at the Foundation for Research and Technology Hellas in Crete, Greece, has throughout her career used this and other computational tools to explore the functional role of dendrites. In the podcast, we talk about what dendrites are, the difference between passive and active dendrites, the kinds of filtering of synaptic inputs the different kinds of dendrites can do, and the key role of the detailed placement of synapses on dendrites has for determining their functional role. Multi-compartmental modeling can essentially be used to model electrical properties of neurons with arbitrarily complicated dendrites. However, it is computationally expensive, and sometimes simpler modeling schemes are needed. In the podcast, we talk about simpler ways to model neurons with dendrites, including the software tool Dendrify, developed in Yiota's lab. This tool facilitates the addition of genuinely dendritic effects to the popular integrated fire type neuron models. Interestingly, dendrites play no role in today's AI. The simple neural units used in artificial neural networks is described by a single dynamical variable, analogous to a model of a biological neuron where only the membrane potential in the soma is modeled. Towards the end of the podcast, we talk about how dendrites may facilitate AI both in supervised and unsupervised learning schemes and also in low power neuromorphic computing. As for all episodes, information about episode and relevant links can be found at the webpage theoreticalneuroscience.no. If you want to support the podcast, go to patreon.com/theoretical neuroscience podcast and choose a membership of your liking, for example, at the cost of $1 per episode. And as a member, you will get access to a video version of the podcast recordings as well as transcripts of each episode. And note that there is a 7-day free trial period when you sign up. I will be most grateful if you become a Patreon supporter to help me fund the production of the podcast, even if you're not too interested in looking at the videos. But I also appreciate all the other listeners, and I hope that all of you can help spread the word about the podcast to other people in the community, on social media or otherwise. Okay, here is Yiota. So, welcome to the podcast, Yiota Poirazi.
Very nice to be here. Thank you for the invitation, Gaute.
So, you are, I just like a little introduction, you're a computational neuroscientist. I will certainly come across
I would
many times.
say, 95% of the time, because I recently started doing experiments also in the lab.
Wow.
But yeah, I think of myself as a computational neuroscientist. Yes.
That's very brave. So to do that at the same time, I wouldn't have the nerve for that. But
It's
anyway,
fun.
or the skills. So that's good. And this podcast is going to be about dendrites, and of course, you are. I guess over the years, your particular focus has been on dendrites and what they can do in terms of computations.
Absolutely. We are a dendrites lab.
Dendrite lab, and your lab is at. I just found you on the web that you are at the Foundation for Research and Technology Hellas, called Fourth, in Crete. It's a beautiful location. I was there last year, so it's really a nice place. So you have sort of studied. I think you took your PhD in Southern California, working on dendrites
Yes.
back in the days, and so I thought we should just start with what are dendrites?
Yeah, excellent question. So dendrites were first discovered by Ramon y Cajal many years ago in the
Mm-hmm.
1880s, 1800s, and essentially they are extensions from the cell bodies of neurons, elaborated and very beautiful extensions. And at the time, they were essentially thought to bring nutrients to the cell body. Later, we figured out that they're essentially the main site of incoming inputs to the neurons. So over 90% of the synaptic connections between neurons take place within dendrites. So
Mm-hmm.
now we know they are much more important. They provide the surface area for connections, but they can also do very fancy computations, which I guess we will discuss today.
And they're probably the most beautiful thing in neuroscience. I mean, they really are. I didn't sort of like Carl call them the butterflies of the something butterflies of the soul. No, maybe that was
Yeah,
one
yeah,
more.
I think the butterflies of the soul. Yes, yeah,
That's a
they're
really beautiful poetic way of saying it. So,
indeed
so I mean, now a main function is sort of to be like the to provide. Well, it's like a way for where presynaptic neurons sort of provide inputs to like postsynaptic neurons, which then sort of process this information and maybe provide then the output in terms of action potentials from its own soma via the axon. So, I mean, I think the study of dendrites that was like the pioneer here was really Wilfred Rahl
correct.
in 50s, 60s, and he in particular studied passive dendrites. So, what are passive dendrites?
Correct. So at the time of Woodford trial, we didn't know that there were active ionic conductances in dendrites. And by active, we mean channels whose opening is dependent on voltage, like the channels that underlie the action potential, for example, the sodium and potassium channels. So back then, we did not know they also existed in dendrites. And if you don't have such conductances, which are termed active, everything else is described as passive. So
Mm,
essentially, the ability of signals to propagate from the site of their initiation in the dendrite to the cell body in a way that is not influenced by such conductances. So
mm,
they travel and they attenuate, and this attenuation depends on the morphology of the dendritic tree, for example, the properties of the membrane to store charge like a
mm,
capacitor, and also how leaky this membrane will be, so how easy ions would flow out through the
mm,
membrane. If
mm.
we only have these kind of properties, then we term the dendrites passive.
Yeah. So, and I guess one aspect of aspect of that or consequence of that is that these passive dendrites are linear in the sense that if you double the input current to you get twice the double the response, right? So
So
that's like
that's not exactly accurate. I mean, this is the case for relatively small currents.
mhm,
So if you look at the integration curve of passive dendrites, it is actually sublinear. So it starts out being linear, close to the linearity. So if you give small amounts of currents
mhm,
and then double them, you do get double the response. But as
mhm,
you grow into bigger and bigger currents, primarily because of the leak conductance, they start saturating and you have a sublinear response. We still model them using the linear cable theory. And I
mhm,
guess that's why people have thought of them as linear, but in reality, passive dendrites are sublinear,
mhm.
at least to my knowledge, right?
Okay, yeah. So that's because it's also one additional nonlinearity comes from the. I mean, if you inject currents with an electrode, then it's like no. I mean, then well, if you instead do it via conductance-based synapses,
Hmm.
so then you also have a non-linearity from the because then the current depends on the distance from the reversal potential of the cell, right?
Absolutely.
So then
Changing
you
the
get the
driving potential.
yeah. So
Yes.
then you also get the non-linearity that way. But at least if you can sort of in the assumption under the assumptions that you so like this linear approximation is good is applicable or reasonable, then it's like easier to model because then you can do it's a linear system, and then we have all the techniques from physics with Fourier analysis and things. So we have benefited a lot from that in
Yes, absolutely.
our work. So okay, so then, but then of course that's more like from a modeler's perspective.
Mhm.
But another kind of, a more important thing from the neuron perspective is sort of what kind of filtering of synaptic inputs can linear dendrites do, or
All the
passive dendrites?
yeah,
Passive dendrites.
they cut off the high frequency inputs,
Yeah.
so they serve as low pass filters, obviously,
Mhm.
and
Yeah.
that is important for a wide array of computations. So even if the dendrites do not have these voltage dependent conductances and they act
Mhm.
in this manner, they can detect low pass events, so they can implement specific computations.
Yeah. And then, so that's sort of this low pass filtering comes from these RC properties, or like resistive capacitive properties that you mentioned. And but then there's also, this what we call sublinear integration is also a feature of these.
Yeah, that's
So,
exactly what we mentioned before, right? The sublinear
oh,
integration, where you have multiple synaptic inputs coming in,
uh,
and where you would expect that as you increase the number of inputs,
mm,
the response would increase in a linear manner, but this
mm,
doesn't happen. And
okay.
that way they are thought of as sublinear integrators.
Uh,
And we have evidence of such sublinear integration in cerebellar neurons, for example, but also in pyramidal neurons in thick
mm.
apical trunks. There are some really nice old studies from Cash and Yusti, for example, back in 99, where they reported that the apical trunk of pyramidal neurons integrates in a sublinear manner.
So it means that because one effect that if you have just like a single, if you have passive dendrite and you move the synapse away from the soma, the peak voltage or the peak current, it will be low pass filter, so the peak will change. But this is something, now you're talking about the sublinear integration of several inputs. So that's another
Yes,
effect.
yes. So, yeah,
That's
right.
another effect.
That's another fact. You were talking about the attenuation. So the further away the signal, the more
Mhm.
it will be attenuated until it reaches the cell body.
Yeah.
I guess we're confusing the integration mode here.
Yeah, exactly. It can sort of mean, yeah, exactly. So the thing is, okay, so okay, just to summarize, because passive dendrites are so much there, you actually, even gentlemen like me can sort of get some kind of principles out, right? That you have the further away you are from the soma, the typical a single input gets dampened more, and you lose low frequencies.
Right.
But in addition, if you have more than one input, they also the net effect is not just it's sublinear summation, so you don't
Yes.
get the sum. Okay, very good. And of course, this and one effect here is also, since the sequence of not only the distance but also the distances of different synapses, but also the sequences
You
of
mean the
synapse
temporal order, right? The
temporal order.
order.
Yeah, temporal order also
Yes.
matters, right?
Yes, yes, absolutely. And even Woodford Rall noted this as an effect, which
Mm,
depends a lot on the diameter of the dendrites, because they start thick close to the trunk and they become thinner and thinner towards the tip.
uh,
And therefore, because of the effect of the attenuation of the signal that depends on
mm,
the diameter, if an input comes
mm,
inwards from the tip to the dendrite versus coming outwards
yeah,
from the trunk to the tip, you will have a very different response. And passive
mm,
dendrites can actually be used to detect the order of
mm.
stimulation.
So, because your group and our group, you have working with dendrites, and I sort of been often struck by myself or in our group, and we sort of even understanding the properties of passive neurons and how they behave. Often we are sort of, oh, it must be like this, and we test it in a simulation. Oh, it's not like that. It's sort of just understanding the full aspect of what a passive and then, like dendritic structure can do and effects of functions. It's not that trivial.
I fully agree. And in fact, I don't know if you're familiar with a very nice work by Romain Cajal and Boris Gudin, where they studied mathematically
Mm,
what kind of logical operations can passive dendrites solve, so
mm,
non-linearly separable problems. And
mm,
they concluded that in fact you can solve as many types as with a sigmoid or an active dendrite, but you just need many more of
ah,
them and to
I see.
combine them in
Yeah,
interesting ways in deep
yeah,
networks.
yeah.
So a passive dendrite is a very, very powerful dendrite. It's not
Mm.
something to think of as a insignificant feature of a
It's a
neuron.
advice to young researcher, at least if you want to go into sort of like the biophysical biophysically detailed modeling to really take some time to invest in and learn these things, because it really is sort of I just remember this story. It was actually the son of Niels Bohr. He was like Bohr is sort of like this famous Danish atomic physicist, and his son was asked, why was your father so good in physics? And he said that, well, it's not that really my father understood all these fancy new sort of quantum field theories and this thing that came. It was just that he really understood the basics of physics,
Mm,
and not
mm.
many people do that. And I think
Yeah.
it's sort of not many people in the field really understand the basics of passive dendrites. And I think
Yes.
that's because
Well, I would say that not many people in the field care about dendrites,
yeah,
and that's where the problem starts, right?
that's true. Yeah, that's another type of problem.
Yeah.
But even if the ones that they would like to sort of think about dendrites, they should make some investment,
Hmm.
even young and you're an eager brain, to learn about passive dendrites. And there's good books about it, right? I mean,
Yeah.
you have like that, yeah. So, but then so much about passive dendrites. So now, now we're going to really make it hard, right? With the active conductances,
Mhm.
or well, I guess you call them active dendrites. Do you call them that?
Yeah, we call them active dendrites. And essentially,
Hmm.
what we mean by that is that in these dendrites, we have conductances whose opening and closing properties depend on voltage. So
Hmm.
they don't have fixed opening
Hmm.
and closing conductances, primarily voltage. That's the ones we deal
Yeah.
most with, at least in my lab. And
Hmm.
the most known ones are the sodium and potassium channels that underlie the action potential. But
Hmm.
there are many others, calcium conductances, different types, N type, T type, R type, you name it. Um,
So
what else?
yeah, exactly. So I mean, it can depend on voltage, as I said, and or even ion concentrations, and of course it's time dependent. And I just looked up in a book. It is like about 150 different sort of channels like this in the humans, used in humans. So it really is a zoo of just different elements, right?
Yes, yes.
So the space of like possible neuron models are enormous. I was thinking, if you have like a very simple neuron with only 10 compartments,
Mhm.
and each of them only get one ion channel type, which is sort of completely unrealistic. But even with a fixed density, so just one number, and even like with well, I mean, if you have 10 compartments and 150 different types, right? You end up with just with that, more combinations possible than like atoms in the universe, right? Or
Right,
particles in the universe.
right.
So it's just like but there is so at least I remember I started joining. I joined neuroscience as an adult. I would say I remember it was not something I got in my during my undergrad years or something. I studied physics, so I just remembered I was just overwhelmed by this complexity of ion channels, and I didn't
Mhm.
really know how to think about it. We learned about the sodium and the potassium that underlies the Hodgkin-Huxley kind of action potentials, but beyond that, it was just too much to take in. but but now I've learned that there is some system to them, right? You have, I mean, one class is regenerative conductances. So
Right.
what are those?
Well, regenerative conductances are those that generate events that are regenerative, like spikes. So we
Mm.
can have sodium spikes, pretty much the same, maybe slightly different than the ones that we have at the cell body, than the action potentials generated by
Wait
sodium
a minute.
conductances.
Somatic spikes in the dendrites. Mm.
Yes, something that looks like a soma spike in the dendrite. And then we
Hmm.
have calcium spikes mediated by
Hmm.
calcium channels, but they're typically wider and more long lasting than sodium
Hmm.
spikes. And we also have NMDA mediated spikes.
Hmm.
So those are underlined by the current that goes through the NMDA receptor, and that's mostly calcium channels, calcium ions underlying that, but it's a different shape of a spike than the typical calcium spike. And we
Hmm.
even have the calcium plateau potentials,
Hmm.
which are also calcium spikes, but they're typically much longer in duration. Let's say a calcium spike would be 30 milliseconds, a plateau potential would be 100, 150 milliseconds. So we have at least these four types of spikes taking place in the dendrites.
Hmm. And they are all. I mean, the reason that calcium and sodium is involved or are involved is that they have like a higher reversal potentials or positive reversal potentials. So whenever these channels open, they sort of you get the cell depolarizes from the resting potential of minus
Exactly.
70. So
Exactly. And
so
they're also tightly controlled inside
yeah.
the neuron.
So instead of at least when I started thinking about sort of like not sort of like the different names of the channels, but really the ions that are involved. So calcium and sodium,
Hmm.
they are sort of like the one like we're going to depolarize the cell. So they are typically involved in like regenerate. They are sort of regenerative conductances.
Yes.
And but then you also have NMDA spikes,
Hmm.
like you mentioned then. So they're so why are they regenerative?
Well, because they are different from the calcium spikes because they don't go through a channel. They go through a synaptic receptor, which is the
Mhm.
NMD receptor. This receptor, in order to open, it has like a two-stage thing. So it needs the glutamate release and it also needs the magnesium, which is blocking the receptor, to be removed so that it opens. But in the end, it still passes calcium channels, right? So the underlying current that goes through the NMD receptor is a calcium current.
Okay, so
So
it's
in that sense, it is the same thing. It just goes through a different machinery, if you like.
but this regeneration thing, meaning that if you get over a threshold, it starts feeding to itself. That
So
has
yeah,
to do. That's an NMDA thing having to do with the magnesium, right?
exactly. So to pass the threshold for opening the NMDA
Ah,
receptor, you have to remove the magnesium plug. And this removal is dependent on voltage. So
mm,
first, it has to be a depolarization that can be achieved by current going through the AMPA receptor, let's say at the synapse.
mm,
Then it gets it to a point, remove the magnesium block, and then you have the current through the
Because we don't
NMDA.
talk about AMPA spikes,
Well, we
or do we?
don't.
No,
Although we should,
we
because
should.
yeah, we should. I mean, in the sense that they also drive a depolarization, which is much faster,
Uh,
right? And it
mm,
returns to baseline with a much faster kinetic than the NMDA spike. But I guess probably because it is so fast, you're right. Why don't we talk about AMPA spikes?
yeah, mm.
No, okay. So the reason why we don't talk about AMPA spikes is because why don't we? They don't get regenerated. So
Yeah,
it
because I
is
think at least the magnesium block is not there. I think that is sort of like something
yeah,
that
but there is no magnesium block in the sodium spikes
yeah.
either, right?
Then, but at least yeah. No, I don't know. So yeah.
I think because in order to have no, but it's the same. In order to have an AMPA opening, you need the neurotransmitter release. But this is true for the NMDA spike as well.
Mm, mm.
Yes.
But at least at the different at levels of conductances, the main, at least from my sort of my modeling point of view or what I've learned about them, I'm like, I mean, in MDA spikes, you have this magnesium block that is has to do with that is released when it's depolarized sufficiently. While for AMPA, it's the current is
Yeah,
no,
this is the voltage dependence, let's
yeah,
say, of the
yeah,
mechanism. This voltage dependence is there for
and
all of these regenerative events, and it's not there for AMPA because
yeah,
AMPA
maybe
does not depend on the voltage of the membrane in order to open. It only depends on the neurotransmitter release.
yeah, or in
So
the
that's a big difference.
yeah, maybe that's it. Anyway, so but then now we're going to come back to the functions of someone like this is like calcium spikes and so on, what they are. But first, I want to just get like the players up on the table. So we have regenerative currents, and then you have restorative currents. That's
Right.
another class. Mhm,
Very true. For the most well-known one is the H type of current,
mhm,
which is activated at negative potentials, and it has and its
yeah,
major role is to restore the membrane back to its original state. So, if you have a strong hyperpolarization, you want to return the membrane to minus 70, which
mm,
is a typical resting membrane potential of the neuron, and
okay.
that's why we call it restorative. It wants to restore it back.
Yeah, so that's the one that sort of if you sort of exactly. So you're saying if you are at rest, let's say minus 70, and then you get really sort of hyperpolarized to minus 80. The IH helps you
Back.
to come back, right?
Right.
Yeah.
Exactly.
So this is sort of like, but I guess in general, all there's also this potassium currents, right? So they also because potassium has uh, well different from sodium and calcium, potassium has sort of like a reversal potential lower than
It's a hyperpolarizing current,
yeah,
potassium.
exactly.
There are
So that
many of
also
them,
yeah,
right? There are
so
many
that
of them.
also often acts sort of like yeah.
Well, not restorative. For example, if we look at the A-type current, which
Okay.
is a very well-known current, and it has a different distribution in the proximal and the distal dendrites, at least, of
Okay.
pyramidal neurons. One of their important role, for example, is to sharpen the after depolarization. So after a spike is induced, to sharpen the return of the voltage to the membrane,
Okay.
because they are activated just after the spike. And because of this effect, they can also kill some spikes. So you can have a spike blocking effect because of the A-type current, or you can prevent the backpropagation of the spikes into the dendrites because of their activity.
So that's also restorative current, right?
Well, if you think about it that way, because it's
Yeah.
trying to bring the membrane
Back.
back to
Yeah.
minus 70,
Yeah.
yes.
Yeah.
Yes.
Oh, I just want to have some simple pictures in my head, so I'm not. Yeah. Anyway, so I think you have like 25 years to sort of to get all this information integrated into your brain. So
Yeah, and I have to say it's not always straightforward, right?
no.
For example, one type of current that I find fascinating is the slow after hyperpolarization current, which
Mhm.
is a calcium induced potassium current.
Ah. Yeah.
Yes. So
Yeah.
if you have a lot of calcium, for example, at the cell body, and you have some bursting, then these currents kick in, and they are more and more activated the more calcium that you have, and they restore. They're also restorative. They restore the membrane back to the minus 70. And now, if you have a small there is a competition between the calcium that activates the calcium channels and the calcium that activates the potassium channels. So they could be fighting to keep the membrane around a set point, and that I find fascinating.
Yeah. Exactly. So okay, so what? Okay, so then we can start talking about the function of all these active dendrites and one active conductances. And one, I think I remember I was fascinated when I read about this about some years ago with this, that actually the active conductances can sort of contribute to what something called a synaptic democracy.
Hmm. Hmm.
Yeah, what is that's sort of a
Okay.
cool name. What is synaptic
Yes.
democracy?
Okay, so these are a few studies. I don't really agree with synaptic democracy, to be honest, because I think
Okay,
that active conductances are there for something else. But I
okay,
will explain. So
uh,
if you have synaptic inputs that arrive in locations that are very far from the cell body,
mhm,
because of the passive propagation and the attenuation of signals, their effect at the soma will be tiny compared to inputs that are very close to cell body, right?
mm,
So there will be disadvantage. They have a huge disadvantage, let's say, if they're very far. Now, how can one overcome this limitation? You can
mm,
place some regenerative conductances, active conductances towards the tips or far away from the soma, so that you boost these inputs in a
mm.
way that whatever arrives at the cell body is, let's say, has the same effect independent of location. And
Yeah,
this is what we call
yeah.
democracy,
It
synaptic
sounds nice,
democracy.
but you don't trust that that's what the neurons
Yeah,
really are
I
doing.
don't think that the neuron will go onto all this trouble to come up with active conductances if
Yeah,
it was only going to use them to linearize things, because you
yeah,
can do so much more with these nonlinear conductances
that's
that
true.
just allow the inputs to
Mm.
have the same
Yeah,
effect.
yeah. But maybe there's some advantages to. I mean, like I guess if you just want to do some signal processing, then linearity is an advantage, right? If you make a decision, it's obviously
Well,
non-linearity.
it's linearity is advantageous, let's say, for us because we understand it much more
Yeah,
easily and we can
yeah,
mathematically describe it. I'm not
yeah,
sure that in a system that has the ability to do more advanced nonlinear computations,
yeah,
linearity is a major gain,
no,
especially if you get it by coming up with all these expensive mechanisms
yeah.
of distributing channels in precise,
Mm,
amounts
true.
as a function of distance simply to achieve linearity.
Yeah.
So
But I guess if linearity somehow is also useful not only for the modelers but also for the neurons, then of course with it you get much more place, no, bigger branches to you can attach more synapses.
yes, yes. And that's one of the major contributions of having dendrites, right?
Yeah.
To
So
increase the surface area of the
yeah.
neuron without increasing
So
its volume so that you can have as many possible inputs
mm,
as you can get into a neuron. And certainly, so it could be that let's say, one of the benefits of having active conductances is to have some linear summation if that is desired, but
mm,
I bet it goes much more than that.
yeah. So so now beyond synaptic democracy. So one idea is that this, I mean, these calcium spikes, at least sort of the way I sort of learn about them, is that there's some kind of typically, well, at least they can sometimes start in the soma and then back propagating spike, and propagate up the apical dendrite, and then maybe then, and this can be maybe this, then there they maybe can, well, they can meet sort of like apical inputs, and I guess that is sort of an idea about this coincidence detection, right?
Right. So the calcium plateau potentials, which are believed to be generated in the distal apical tuft of pyramidal neuron in response to apical input and coincident backpropagating action potentials, they are thought of as predictive signals. So indicating at the neuron that whatever comes in as an input from the apical tuft is relevant to what comes in as an input from the basal tree through the generation of these large calcium plateau potentials that then drive bursting activity at the soma. And this bursting activity is thought to be a signal for whatever coincidence detection or driving plasticity at the feed-forward synapses so as the neuron to learn to identify these coincidences.
Yeah. So
That's one
yeah,
hypothesis.
exactly. So this is sort of like this. I mean, this is this predictive coding
Mhm.
ID, right? That they have some expectations coming in at the neurons apical dendrite, and then meeting sensory input coming from the soma or
Yeah,
something.
that's one theory, right?
Okay.
Because the mechanism itself was observed by Matthew Larcombe back in the 1999, I believe,
Mm.
and it was done in vitro. So we didn't know anything about expectations and feed
Yeah.
forward and feedback, the meaning of these signals, let's say. But based on these observations, people have proposed the predictive coding hypothesis.
Mm.
Yes.
Cool. Yeah, I guess this predictive coding idea came around like also late 1990s, about the same time, or at least I don't. Yeah. Anyway, and then but there's this other thing that now you can also have this like NMDA spikes to help in coincidence detection of. Then I guess if it's different synaptic inputs entering at the apical dendrite.
So there are different things you can, yes. So there is some really nice work back from the lab of Jackie Chiller, and then later on from many neurophysiologists, including Jeff McGee and Attila Losonci, where people have shown that if you have a small number of synapses activated together in time and space, then they produce some depolarization that is enough to remove the magnesium block and then open the NMDA receptor and then drive a large NMDA spike. And this would be a way for a dendrite to detect continuous coincident inputs. To have that, you need to be temporally precise, so within just a few milliseconds. And this is required to drive the sodium spike, which then would open the NMDA spike. But
The sodium spike up in the apical dendrite.
yeah, any dendrite. In fact, this was shown in basal dendrites, in oblique dendrites, wherever
Okay.
you have sodium and NMDA conductances, you
Yeah.
can capitalize on this property.
Mm-hmm.
And if synapses are spatially close,
Mm-hmm.
then the temporal summation of the APSPs that are produced is more effective. And that's why the idea of clustering was also emerged back then. So if the closer you put synapses together, the fewer of them you need to have enough voltage to surpass the threshold for a spike. That's
Ah,
the idea.
yeah. So, I mean, this is, and this typically, this short time scale for coincidence detection, if that's what you're interested in, that is much easier to get up in the, like in the thin, say, apical dendrites than if you do it in the soma, for example.
Yeah, you couldn't really do it in the soma. First of all, you don't have an MDA receptors in the soma, right? The synapses are in the dendrites. So,
Yeah.
but it also wouldn't really make sense to have it in the soma, because the reason it makes sense to have it in the dendrites is that you can have multiple dendritic units operating simultaneously in parallel, detecting such coincidences. So at different time points, you can have different dendrites or even parts of a dendrite detecting this coincidence, signaling one input, and at another time signaling a different input, and you have a parallel computing machine, which is what we proposed back in 2003 with our paper with Bartlett Mel.
Mm.
So that's why it makes sense to have it, in separate dendrites instead of lumping these things at the cell body. And I guess that would be one reason why biology doesn't have a lot of NMD receptors in the cell body, right?
Okay, so I mean, we had we had an earlier podcast with Christoph Koch,
Mm-hmm.
which wrote this seminal book, Biophysical Computation, back in 1999 or something, and I guess there he was one of the reasons he wrote that book was that his interest in this, what is sort of like the so identifying possible biological substrates of particular logic gates, right? Maybe like motivated by I think like today's computers is that like not and or something, which is
Yeah,
one
logical
of the
gates, yes.
logical gates. But I guess there's all kinds of possibilities for various, for at least for some key logical gates also in the dendrites.
Absolutely. I think, I mean, that old work by Christoph Koch and Thomas Poggio already showed that with the dendrites, essentially, you can solve
Mm.
almost all logical gates, right? That was done primarily with combinations of multiple dendrites, not just one. And they have shown that, for example, the NMDA receptor may act as an AND gate, because if you have multiple inputs, both of them together, then you surpass the threshold and you induce a response, and that's an AND
Mm.
gate. And recently, together with Matthew Larcombe, we had a paper where they discovered a new type of spike in human neurons, later also reported in rodents, that had a non-monotonic activation function, not activation function, sorry. Its amplitude was changing in a non-monotonic manner in response to the strength of the input. And in that paper in Science, we predicted that because of this non-monotonic function, dendrites can also solve the exclusive OR
I see.
computation, which is a challenging one and for which you need a small network of neurons to solve. And I should also point out here that there was a paper that I did not know about back in 1992, in fact, by Zador, a conference paper where he also did some modeling work and put some high conductance of AHP currents that we discussed before.
Mhm.
And because of the interaction with the calcium channels, this reverberating interaction, they also showed that you can solve the XOR. So it
Mm.
seems like if you have particular type of conductances in the dendrites, they are capable of solving very complex logical gates. And what is really impressive is that even a single dendrite can do these highly difficult computations like the XOR. So I think that is fascinating.
Yeah, so there's not a lack of sort of possibilities for how how sort of like logical gates can be implemented, right? I
Yeah,
mean, there's like several.
absolutely.
Yeah, yeah. And then you also mentioned that each branch can be almost like they have to do their own little computation and
Hmm.
maybe meet in the soma, like if you have
Yes,
different basal dendrites, and so that sort of that sort of increases the potential certainly for doing computations.
that's like a deep neural network, if you like, where the number of layers would correspond to how many independent subunits you have in a neuron.
Mhm.
And if you consider the dendrite an independent subunit, then you would have, I don't know, 50, 80, whatever you have in a pyramidal neuron. If you consider a part of the dendrite, so just a stretch of a few synapses, clusters, for example, then you have many more such nonlinear subunits. And of course, as we know that artificial neural networks, which have such layers of independent units, are universal approximators. So
Mhm.
you can solve any, essential theoretically, any problem with such a network. You can think of the neuron also like a deep neural network, and therefore you have a much higher storage capacity and memory capacity, as we proposed in some of our older work with Bartlett Mel.
Hmm. Okay, so now we talked about sort of like I guess, at least, I mean, the role of sort of like regenerative conductances in making spikes and test for like coincidence and maybe like this ideas for like predictive coding, how the somatic input and the apical input can sort of be compared in some sense, or at least processed together. But what about this? You mentioned this IH channel. What's the point of that? Well, it's restorative. Maybe that's all right. It keeps the neuron from.
Well,
Can
I
it?
mean, you
Can
know
it? Does
better,
it do
right?
any?
Because you did some really nice work on that.
Well, yeah, we looked at the extracellular potentials involved in it, like that. If you have IH current, then sort of what would the LFP and stuff like that look like? But we haven't. I mean, the work and what the function could be
Yeah.
is that's a different thing.
Yes, right. So one of the studies of the functions that have been attributed to IH is to block the non-synchronous inputs. So everything that is non-synchronous is like, cut off because of the IH that favors high synchronicity.
Excellent. Yeah, cool. So it's not.
Of course, this can also be something that is achieved through the generation of sodium spikes in the dendrite. What I mentioned before is that if you have very tightly activated synaptic inputs, then you can also effectively drive sodium spikes, followed by NMDA spikes, and that could also be a coincidence mechanism. So there are multiple, I would say, ways that you can detect coincident inputs in dendrites.
And one thing you have sort of worked on is sort of like the what you can do with like essentially that the positions of synapses are crucial, right? You can you can sort of, even if you have a set a set of synapses and a certain dendritic structure with conductances, you can do different things depending on where how you say put these synapses, right?
Yeah, it all comes back to the same idea. So the idea is if you put synapses close to each other, they cooperate and they summate in a nonlinear manner. They are the resulting EPSP summate in a nonlinear manner. They drive a local spike, and therefore you have a nonlinearity. If now you separate the same number of inputs far apart into different dendrites or distances where their resulting depolarizations no longer interact, then you can have a linear or even sublinear summation of these inputs. So effectively now the neuron not only detects the number or the strength of the inputs that come in this way, but it can detect their arrangement, because one arrangement would give a very different result from another. And this also greatly expands the computational capabilities of the neuron. And it also now means that what we know as a synaptic weight, so the strength of a synapse, is not an independent variable, is not a number, because it depends on which is its neighbor. If its neighbor also fires, the effective weight of the synapse becomes much larger than if the neighbor is no longer firing. And this is a property that we hadn't really thought of before. And it's really a smart way, let's say, of the brain to capitalize on the nonlinearities in the dendrites.
So it's but it's like a new level of complexity.
Exactly. It's a
Yeah.
new level of
And
complexity.
possibilities, of course, but also confusion. Possibilities for confusion. So I think you talk about like dendritic subunits. That's sort of like a term you've used. What is a dendritic subunit?
When I say mention something is a subunit, I think of it as doing pretty much the same computation that the cell body is doing, which is having a threshold and producing a nonlinear sigmoidal response, a spike. In this case, I'm not differentiating which kind of spike. It just has a threshold at nonlinearity. And I call it subunit based on the work of artificial neural networks where we have multiple nodes. And I think of a dendrite as another node. But it could be so much more, because you have neuromodulation, you have plasticity, you have a lot of other effects into neurons that could affect how a particular chunk of a dendrite is integrating information. And under one condition, you could have a subunit that has a particular activation function, and then 3 hours later this particular activation function has changed because of neuromodulation or because of plasticity. So I can think of the neuron as essentially having a very large number of potential subunits that go beyond the number of physical subunits, which would be a piece of a dendrite, if you like. Another level of computation, of
Yeah,
confusion.
confusion. Yeah. So, and because I think you showed back in the days that that obviously the a key idea in neuroscience is that memories are, well, at least partially stored in synapses.
Mhm.
And I think you showed back in the days that you can store more information in synapses if you have like some kind of subunits that behave
Yes.
non-linearly.
Yeah, exactly, exactly. Well, I mean, it kind of makes sense, right? Because you are comparing a perceptron, so a small linear network, with a two-layer artificial neural network, which has
Mm,
double the number of computing units, and in that
mm,
respect, you can store
mm.
much more information. So if you have in the dendrites acting as independent subunits, that's why you can process more information. And we actually analytically measured the amount of information that you can store if you have these subunits back in
Mm.
2001 with Bartlett Mel in one of our paper. But more recently, in 2016, I believe we had a modeling paper where we simulated dendrites with active nonlinearities and applied them on multiple memories that are separated across time. And we show that if you have these clusters of synapses within dendrites, that is a very effective way to create associative memories. So
Huh.
to bind pieces of information. So a few of the synapses could represent, let's say, one piece of the memory, the sound, and the other synapses would represent the food shock that we normally have in experiments in animals. If you want to associate the sound with the food shock, it is very beneficial to put these synapses close to each other so as to create a small cluster that will fire a dendritic spike and signal to the neuron that what I have here is a strong association. And we did some simulation work where we showed that if you have this clustering, then you can much more efficiently, which means with just fewer neurons, create such associative memories.
Huh.
So there are two
Mm,
benefits of these nonlinearities, at least.
mm.
One is that it's a very easy way to bind information in space and time to create associations. And the other is that you extend the capacity of your network by making it able to store many more memories.
So, I mean, I guess when I look at the sort of the, well, dendrite or dendrite literature, a lot of work has been done on pyramidal neurons, typically from hippocampus or neocortex. And so, how much does this or your insights transfer to other neurons? You think, like,
Yeah, that's a very good point. Not that many people have studied other cell types. I guess it has a lot to do with the lack of experimental data on many of the interneuron subtypes, for example. In fact, in my lab, we did do a model of a PV fast spiking basket cell a few years back. And we, according to our predictions, they are not that many differences between pyramidal and those inhibitory subtypes because we also predicted the occurrence of dendritic spikes in these basket cells. And in fact, a couple of months later, there was a study from the Kuhlmann lab that reported two types of dendritic integration in these neurons. And now nonlinear dendritic integration has also been reported in SST interneurons.
mm,
So it seems that the dendrites of interneurons, at least these subtypes that have been studied, are also interesting and may have active conductances and may be doing complex computations like pyramidal neurons. But we need a bit more experimental data to be able to build reliable models.
mm,
But we're getting there. I think people
okay.
are interested, and other groups have also done models of interneurons.
Mm,
Francis Skinner, for example. So people are
mm.
getting into this and they're interested.
Okay, so now we're over into to models of neurons, which is of course of key interest in this podcast. So multi-compartment models is sort of, I mean, in principle, you can sort of always model things with multi-compartmental
Yeah.
or
I,
models, right? There's not
as
like a
long
limitation. Yeah,
No, theoretical not at all, but the limitation comes from experimental data that you need to constrain these
yeah.
models. That's the
Mm.
only
But you
problem.
get sort of like typically the anatomy you can get fairly well from this whatever neural lucida reconstruction. So,
Absolutely. And there
so
are a lot of reconstructed anatomies of many different types of neurons available online. For example, in the Neuromorpho
mhm.
database,
Yeah.
the Allen Brain Institute database, even the Blue Brain project has a lot of morphologies available. So there are a lot of places one can find reconstructed morphologies for sure.
And there are also databases of like ion channels, right? Sort of where you can sort of models for different kinds of ion channels. I guess you have
Yes,
Channelpedia
yes.
and what's called ion channel genealogy or something. That's the one I know about. Maybe
Yeah,
not.
yes, from the lab of Tim Voigels. Yes,
Yeah.
that's a very nice one as well.
So
Yes.
now, so you have the building blocks. It's just putting them together and fitting them, right? Or getting where to put the ion channels and
Where and their conductances.
uh,
Yeah.
mm.
Which is not trivial, right?
No, no, that's
Because
true.
these
That's
models are highly complicated typically in terms of the compartments, but also the types of conductances that they have.
mm,
And
mm,
finding experimental data to constrain them all is not easy. Of course, thankfully, we have some automated tools now that people can
mm,
use to constrain these models based on some recordings, some experimental data.
mm.
But even that's not always
But
easy.
and typically these recordings, I mean, these models are typically with from recordings, intracellular recordings in the soma, right?
Exactly, that's the
So
biggest problem
you don't get sort of that much information from the
from the
from the
dendrites.
dendrite itself. So that
Yes,
is still a challenge, or
yes,
it is a
yeah.
challenge. So it used to be that, I mean, there were sort of a handful of. I'm being a modeler myself. Sort of, there were like a handful of models that were around that you could use, and like the main Zinovsky models, and you had also like we lately been using this model developed by Ethai Hay.
Hmm.
So I mean, it used to be like a handful of models,
Yeah,
but now there are like this automated scheme. So now you get databases of model neurons, like with the Blue Brain and Allen Brain Institute and so on. But I mean, the generic problem is that it's very difficult to fit the model parameters.
yeah,
So we have a good skeleton. We understand the cable equation, and that level of description seems to be doing well, quite solid. But it's just
yeah.
fitting the parameters, which is very different than from I used to do like semiconductor physics or simulation, and then the simulation was difficult, but we typically knew the
The
parameters.
parameters, huh?
Yeah,
So
so that's like a new challenge,
it's a
and
different problem, I guess.
it's a different problem. So, but it's also, I mean, it's not like in physics, we typically had sort of quite low dimensional models so that you could find a uniquely best set of parameters if you had experimental data. But that's not going to be the case here anyway, right?
Mm,
Or
no, no, but people do find solutions to the problem of feeding the huge
Mhm.
multi-compartmental models, and essentially, lots of people actually prefer to use more reduced models.
Yeah.
Reduced either in biophysical models or even rate-based models or artificial neural network models. So there are alternatives to the highly
To
complex
yeah.
multi-compartmental models. Of course, each model is better suited for a different question, right? I
Yeah.
would say that the detailed multi-compartmental models are the best model one can use if you're interested in a subcellular mechanism, like a
Mhm.
particular channel type, like a local integration
Mm.
between synapses or ion conductances. That's a question that you cannot answer with a simple model.
Mm.
Um,
But even if you have like this, I know this Hay model was a paper from 2011. So there they had like one model that came out with a fixed set of parameters, but they also had in supplementary a bunch of other parameter sets
mhm.
that was essentially equally good. And maybe that's not maybe that's more like a feature of biological systems.
Yeah.
It makes it robust. There are many ways to implement certain things, and maybe it's sort of it's more about maybe all. It's a question of predicting things also. Maybe all these different parameter sets predict the same, essentially the same things. And
Yeah, I would think that there is a lot of degeneracy, let's say,
Mhm.
between neurons. I mean, we see it also in experiments, in recordings. Not every neuron is the same, right?
Mhm.
They have some overall features that are distinctive of a cell type, but
mm,
the exact combination of conductances and distributions that get you to those features may vary extensively. And this is also
mm,
what we see in computational models. And I
mm,
fully agree that this is a feature, is not a bug, right?
mm, mm,
There are some recent studies, computational studies mostly, that show that if you have such heterogeneity in neurons, this could be an advantage. More robust learning, faster learning in these systems, for example.
mm.
So I would expect that
And
it's
also,
also an advantage in the brain.
if something bad happens to an ion channel, then
You
other
can,
channels
yeah,
can compensate, right?
absolutely,
Yeah.
yeah.
But I guess this is sort of, I mean, this was also demonstrated in this crab stomatogastric thing with Eve Marder's
Eve
group,
Marder,
right?
yes,
Yeah.
yes, she
So they're
was, I
seeing
think, the
this
first who
sort of like in the,
extensively, yep,
yeah, studied
absolutely.
this. So there are sort of and there are, like, as I said, these reduced models. I guess the first one was, or at least that Wilfred Rall really explored, was this ball and stick
Hmm.
model.
Yeah.
Yeah.
Yes. I mean, there were ideas of how to build simple models where you just have
Mm,
a cell body and a dendrite extending from it, and
yeah.
that's why we call it a ball and stick.
Mm.
Um,
Because then you can even get analytical solutions to some, in some
yes, which
cases.
physicists like very much.
Well, yeah, they are exactly, makes us happy.
Yeah,
Yeah.
in fact, these models are still extensively used if you want
Mm,
to simulate a large network of neurons, and
mm,
the computational complexity becomes a problem and speed becomes a problem. These models are good enough, and in fact, they are good enough for a lot of kind of even nonlinear computations like predictive
mm,
coding. For example, many people have done studies with predictive coding where we just have a ball and stick model, two compartment
mm,
model, right? So they can be very useful if they are designed in a way that can answer the specific question that we are interested in. And there is also the Pinsky and Rinzel
yeah.
model, which is
Two
two
compartments.
compartment as well, but
Mm,
fully biophysical, in a sense that you have Hodgkin and Huxley equations there that you can solve. You don't need that. You could also go into something simpler like the leaky integrate and fire models, where you do not have detailed models for distinct conductances. So there are a lot of solutions, let's say, to go from the detailed, the multi-compartmental, to something simpler, and a lot of
mm,
benefits.
mm. And you have yourself, your group has also came out with this tool or program, Dendrify.
Dendrify, yeah.
Yeah, I just that came out a couple of years ago, maybe. This I just read about
2023, last year,
yeah,
in fact.
last year a
Yeah,
paper. Yeah,
yes.
what is that? What is that? What
So
is
this
Dendrify
was
about?
interesting. I asked a student of mine, PhD student Mihalis Bangalos, to build a model of the CE3 so that it can study pattern completion and
Mhm.
separation, and he did not want to deal with the complexity of the compartmental models. So he thought, but we are interested in dendrites, right?
Okay,
So he could not just
so
go for
maybe
a
he
ball
was a
and stick.
physicist. Maybe he was a
Yeah,
physicist.
exactly. So he figured, can I find a simpler way to make my life easier than I can study dendrites and dendritic nonlinearities, but not have to go to multi-compartmental models or the integrate and fire models that typically don't have dendrites. So he came up with Dendrify. So
Mm,
Dendrify is essentially something between the integrate fire and fire models and the multi-compartmental models, where you have the cell body is just simulated as an integrate and fire, so one differential equation, and then you can add compartments, as many compartments as you want, but typically you have to keep them small because that's the whole point here. And inside the dendritic compartment, he injects events, which events simulate the spikes that you have in the dendrite. So you have for example, for the sodium spike, you would have a positive current. We call it event because it has a
mm,
square
mm,
pulse, so it's not really, a very realistic current. You have a positive and a negative just after it, and therefore you can mimic the sodium spike. The same way you can mimic the NMDA spike or the calcium spike with different temporal characteristics. So it's
mm.
very lightweight, let's say, to
Yeah.
simulate. It runs very fast. It even runs in an iPad. And it can replicate a lot of the characteristics that the dendritic model can do. We have backpropagation of spikes, for example,
Mm,
through these event-based mechanisms. We can have coincidence detection of distal inputs and proximal inputs driving a bursting response at the soma. So you can do a lot of stuff that you cannot do with point neuron models, and you would need a multi-compartmental one. But now it's very cheap to do it, and it's very fast.
mm. So, but what is sort of then? when you do this reduction and you add this like templates for different events, what is the target, or well, what is some sense what is optimized? It's in terms of what the model should do. Is it some kind
So,
of
okay, so the Dendrify framework is not trying to optimize something. It is there as a tool to build these kind of models bottom up, so from scratch. So you design your neuron the way you like it, and then
mm,
you validate it based on the same way that you would validate a multi-compartmental model. So you use experimental data and try to fit the much fewer parameters of the Dendrify model into your experimental data, but it's not optimizing some kind of function
no?
at
But
the
okay, so
moment.
but it's sort of fitting some sense the membrane potential in soma, for example, and the spiking output of the soma or something
Yeah,
like
but
that.
those are like 7 parameters, so it's very easy to do it. So you don't really need a fitting tool to do it. These are just 7 numbers that you put in there, right? So
Okay.
it's very easy. But we are going to release very soon another tool which is called dendrotwix, and there you can upload the morphology and upload a set of mechanisms and reduce them while keeping the functionality that you want.
Okay.
So that's a new tool. Is not published yet, but there we have some
Cool.
optimization functions in there. uh,
So then the goal is to start with the morphology and then reduce it to something that has the right
Level of
function
complexity, yes, yes.
and function. So it's not really fitting sort of like membrane potential in the soma or membrane potential in the dendrite, or it's like a different,
No,
yeah,
it's
no,
a different. In
different
fact,
different
yeah,
goal in some sense.
it's a different goal. The goal there is to reduce the morphology to something that you can easily port into a large network, so
Mhm.
that is lightweight to run large simulations, and at the same time build your model so that you have a systematic description of the different ionic conductances, because now
Mm.
you, as you said, we have a lot of databases, but the different channels are described in very ad hoc manners with very different
Mm.
equations,
Yeah.
and we want to build
Yeah.
something that is more standardized, and then feed it to whatever experimental data you have, the response at the dendrite, at the soma, take it out,
Yeah.
export it, and go and build large networks if you want.
Mm.
So that's the idea.
Cool. Yeah, because I saw there was a paper last year in eLife by Vibo.
Exactly.
Yeah,
So
so there they have like also automatic reduction, but there the target is more particular electrophysiological data, isn't it?
yeah,
Or
yes. So a big difference, let's say, with their approach was that you have these distributed synaptic inputs onto the dendritic tree, and you want to create a reduction where the effect of this input is preserved, right?
mm,
Whereas we don't care so much about preserving some synaptic input, but allowing the user to reduce at intermediate stages at any level they want, but without preserving some characteristics of the input. For that, you could use the tool by Weibo, which is a very nice tool.
mm,
And there is also a very nice tool by the Segev lab called Neuron Reduce. I don't know if you're familiar with that. We are adopting
mm.
this into our tool because it has
Yeah,
a bit more flexibility. There
cool.
are other approaches out there as well, but not, to my knowledge, nothing that incorporates all the different steps of reduction, standardization, of channels, and also allowing you to tune the model and play with it inside the same framework.
Hmm. Cool. Yeah. No, I think these kind of tools are really important so that we can find some systematic way to make simplified neurons and know what goes into. So it's not completely ad hoc when
Yeah,
you sort of
yeah,
start making these simplified neural models.
yes.
And then of course you have other approaches. I think I mean you had like this two-layer network model that you did back in your I guess PhD student days.
Yeah, I mean, it was postdoc days,
Okay,
but yeah, yes, it
but
was
still
still work done with Bartlett. I
okay,
had left at the time his lab already, but yeah,
okay.
I mean, the idea there was essentially to understand how a pyramidal neuron integrates information, and based on our simulations, we predicted that we should treat a pyramidal neuron as a two-layer artificial neural network, where
Mm,
the dendrites are one layer and the cell body is the other layer. And this was mostly to explain the integrative properties of these neurons. And this idea, I think, has become popular now in machine learning and artificial neural networks, where people now start to think of neurons as not just point neurons, but something that is a bit more complicated.
mm. Yeah. Uh, and I guess another approach is sort of now what I've seen is sort of to actually train these the standard deep CNNs to predict neural responses.
Yeah,
Meaning, so yeah, that's
that's
like a
typically done when people want to, let's say, check whether if you have a recurrent neural network or a typical CNN, whether you can replicate the experimentally recorded responses in the brain of particular
mm,
brain region with the hope of identifying patterns in these networks that you would help us understand how the neuron is solving the problem.
mm,
I'm not sure. We have not used these approaches in my lab,
mm.
so I'm not really an expert on that to comment. But they are, yes, they are very
So
popular as
yeah,
well.
we have actually used it a little bit in our lab, and we have a new paper coming out where, but there the goal is more to if you can sort of speed up the computation so that you can do larger simulations than if you do this, well,
Uh-huh.
it's the standard way. It's done the multi-compartmental
Ah, I
modeling
see.
thing.
I
Yeah, so
see.
that's another that's
That's another
another
angle. Yeah.
that's another angle, right? Yeah. So, but now we're sort of segueing into this very interesting topic, this use in AI, and I guess if you sort of look at this two-layer network model that you sort of developed back in the days, 20 years ago.
20 years ago, yes.
Sorry for reminding you. No, no, that's good. So, but so then, I mean, it seems like if you have like this two-layer network model, I mean, you could sort of implement it in sort of almost like standard AI architecture today. It's just like then the neuron is a way to have a little like, well, I mean, in the biological neuron is more like you need some more, essentially more simple ReLUs to implement it, essentially,
Mm.
right?
In fact, we have a nice paper on that exactly. It's under revision now. I hope it will
Okay.
come out soon, where we did exactly that. We replaced the point neuron in a vanilla ANN
Yeah.
with a dendritic neuron, and a big difference is that in
So wait a minute,
biological
vanilla. I mean, that's like the simplest stripped down version. But ANN, what is that?
artificial
CNN.
neural network, vanilla
Okay.
artificial neural
Okay.
network. So
Okay.
instead of
Okay.
having a node, we have a node with a which has
I see.
dendrites.
Yeah.
So a big difference back in 20 years ago and now is that you don't have an all-to-all connectivity in these nodes, right?
Mhm.
So the soma only talks to its own dendrites, and these dendrites are few, are not that many, and this makes the network way sparser than a fully connected network. And
Mhm.
if you manage to achieve similar performance with a network that has this very directed and more sparse connectivity, then it is a major gain in terms of energy, in terms of area that this network takes.
But the,
Uh,
but
yeah.
these convolutional neural networks that AI people use have like rather restricted kernels, right? So they
Yes,
may
but they are shared with all the new. So the
yeah.
CNN nodes, they have shared weights with all the inputs, and the
Okay. Yeah.
networks themselves are fully connected as well.
Yeah.
So the
Okay.
number
So
of connections in a CNN is massively larger
mm,
than in a dendritic, let's say, network.
mm, mm. So cool. So that's like, and then one thing that you also wrote this nice paper on the connection to AI,
Hmm.
and you talked about, for example, supervised. I mean, now the I guess the workhorse of today's AI are these well learning networks, the backpropagation of errors,
Hmm.
and now there are some attempts to sort of to make to take this supervised learning ID, but to more complicated neurons where you maybe have like maybe two or three compartments or something where there's some kind of trying to impose these supervised learning rules on these more
Yeah,
complicated
I
neurons.
would say there are two parallel approaches. One
Okay.
approach is trying to show that you can implement backpropagation in biological circuits, and
Okay.
people are trying to come up with ways as to what would be the error signal, let's say in a biological neuron. Could it be the feedback information that comes into the apical trunk? Could
Mm,
it be the dendritic plateau potential that is generated? And the feed forward input would be, let's say, the basal input coming in, and there are different approaches by several labs: Walter Sen's lab, Blake Richards' lab, Rui Pontecosta's lab, Richard Knoll's lab, that are coming up with ideas along these lines. And then there is also an approach where people are trying to look at biological learning rules, so unsupervised learning rules,
Mm.
or birth dependent, let's say, plasticity rules that could solve the same problem equally well, but not necessarily trying to claim that this is backprop done in the brain. So
Okay.
both of these
Yeah.
attempts are ongoing right now. I don't know where
Mm.
to bet our money.
Uh,
Personally, I like the biological rules
uh.
more than the back propagation rules, but let's see.
So why? Because you like biology, or because you think they're going to be more efficient in the end?
No, I think that there's nothing more efficient than backpropagation
Uh,
because it's a very directed rule. So, in terms of efficiency, I mean,
mm.
I doubt if we will find something. Yeah, we'll see. We'll see. I don't know. Maybe
No.
we will. But I definitely, given what we know so far about the plasticity rules that are used in the brain, like LTP and LTD, like
Mm.
cooperative plasticity that we know between synapses, plasticity rules that operate at very slow time scales, like intrinsic plasticity of excitability, for example. This is nothing like the typical backpropagation, which is a very, let's say, fast, directed, and going across all layers rule. And I
Mm.
have not seen that in the brain, at least to my
No.
knowledge. I think it is unlikely that the brain
Yeah.
is doing backpropagation. But
Across all layers,
yes,
certainly.
across all
Yeah.
layers. I mean, how? That would be really hard.
But and one problem with the AI algorithms today is like, I guess, transfer learning, or sort of to be able to actually be train a network to do one task, and then if you want to sort of to make it do another task, then it starts forgetting how to do the first task,
Yes,
right?
yes.
Catastrophic forgetting.
Exactly. And that is a problem of back propagation because you update all weights every
Mm.
time the network is trying to learn something new. Whereas in biology, there are some tricks which are actually now being also adopted by computational people to mimic it. Like, for example,
Mm.
in biology, there is indication that if a synapse gets really big, then it's very hard to change it anymore. So it becomes rigid, so it is not easily affected by plasticity. And people have mimicked this property in studies like the synaptic intelligence work by Friedman Zenke, or the elastic weight consolidation. I think that was the other algorithm.
Mm.
Trying to find a way to freeze, let's say, some part of the network, some parameters of the network, so that they are not affected by catastrophic forgetting. And this way
Mm.
you can transfer some information later on or continuously learn in the network. And these are ideas taken from the brain.
Hmm. And there's also some ideas I learned in your paper about sort of using that this NMDA spikes can sort of be serve as a mechanism for sort of like contextual adaptation. I guess sort of like that you adapt your neuron depending a little bit, make your neuron,
Mm,
say like with this like the ReLU neuron, the standard
right.
AI neuron, a little bit different with different thresholds and gains and stuff like
Yeah,
that.
there was a nice
Depend.
paper by Willem Weibo in 2023, I think in PNAS, where
Mm.
they explore the possibility that the NMDA spikes would be a way to learn the context of the input. And the way they would model this effect would be by essentially changing the slope of the ReLU activation function of those neurons.
Mm.
So the effect of the NMD spike will be a different threshold, like a dynamically changing threshold. And they did some really nice work and showed that you can extend this algorithm, this hypothesis into machine learning regime. So
Mm.
that was
Cool.
also quite interesting, I would say.
So, and then, as I mean, a main problem with today's AI is the enormous energy consumption, right? So then there's like this neuromorphic computing is something that we all hope for,
Yes.
and I guess there has been obviously some nice examples of the use of neuromorphic computing, but typically they only had like neurons with somas, right? Point neurons, right?
Yeah, yeah. Although there has been attempts, like for example, the lab of Aridan Bazou, Giacomo Idiveri, and Melika Payar. Now I know they are
Mhm.
building chips, neuromorphic chips with dendritic units. We still, I mean, we still have not solved some of the problems with these neuromorphic chips that have to do with a lot of variability. They're quite noisy. So I don't know how close we are to an industrial, let's say, solution for neuromorphic systems, but there
Mm,
is certainly hope that we can adopt some of the principles of the brain to solve this high energy consumption problem, which is getting bigger and bigger with the
mm,
large language model systems, right? And the brain is so efficient in
mm,
solving problems with 20 watts, right? So
mm,
spiking neural networks, which is what we have in our brain, implemented on neuromorphic software, capitalizing on the dendritic computations, I
mm.
hope,
Yeah,
will be the future.
yeah.
Um,
So
yes.
what's your prediction there? That I mean, you say, I mean, how important will dendrites be in AI?
Oh, I think dendrites will be very important in AI.
Okay.
Yeah. Yes,
Mm,
and I think they will be very important because of the kind of computations that they can perform in a manner that makes neurons more efficient. So if we are
mm,
looking about ways to improve efficiency, then I think dendrites are one of the very
mm,
promising component of the brain that can be used to provide inspiration and new ideas.
mm. I mean, if you look forward to the future, I have no doubt that all kinds of like this the use of dendrites in AI will certainly be explored. There's so much interest in this. Obviously, also commercial interest from big companies, of course, and so we have lots of smart people working there to explore this.
Absolutely.
But what about sort of the more like the biological studies of neurons and dendrites? I mean, you have some are like new experimental techniques on the way. I guess optogenetics is an important way to sort of to explore functions and stuff like that.
Well, I think what is really promising right now is voltage imaging, because
Mm,
now we can in vivo finally keep track of the activity of the dendrites in the temporal resolution that we need to
mm,
detect spikes, to detect nonlinear events. Because until now, with calcium imaging and the very slow kinetics of the calcium sensors, it was very difficult to keep track of what's happening in the dendrites in vivo, right? So
mm.
now that voltage dyes are coming of age and people will be able to study these dendritic dynamics in vivo, I think we're going to learn much more about how dendrites contribute to complex
So
functions.
I mean, if you look at these different techniques, I mean, you have obviously like calcium has been important, and then there you have like particular like whatever markers, whatever like that can sort of can sort of essentially give an estimate of how much calcium there is inside the cell,
Mhm.
and then there is, can you do the same thing with sodium?
They measure the voltage change, not the sodium,
Not the
these
sodium.
voltage sensors. So it's
Mm,
like essentially doing patch clamping in vivo.
yeah. Yeah, but how? What's the? I mean, that has been technique has been in principle been around for some time, but I guess it's more
Yeah,
a
it
question of
had some serious
space.
problems in terms of toxicity, in terms
Yeah.
of the duration for which you could record, but there have been
Mhm,
major improvements over the years. So I think people now, or if not now, in a couple of years, I mean,
mhm,
recently in the Dendrites conference, we had some really nice presentation with in vivo data from Adam Carter, I think, that were quite nice using
mhm,
voltage imaging.
mhm.
So I think we're there.
Yeah, but I mean, then so then you can still have this spatial resolution that you have with this two photon thing, right?
Yes,
Yeah,
exactly.
exactly. Yeah, yeah,
Exactly.
that would be really cool. Yeah.
Yep. Yeah.
So, I mean, I read this or I looked into this very nice book, Dendrites,
Mhm.
that came out in 2009, which has had many nice chapters, and I think it's well, seems to me to be a very nice resource still. I'm not quite. This is a second edition. I'm not quite sure if a third edition is on the way. But
Yeah.
I to get have a book like that that is continuously updated is really a nice resource. Uh,
Absolutely.
and there's uh, I mean this there they say in the end that like the main challenge is to understand the relationship between form and function. So this was written in 2009 or something.
Hmm.
So, how much closer are we to understanding the relationship between form and
Mm.
function of the neurons and
Yeah,
the
good
dendrites?
question. I mean, I think we've made important progress in
Mm,
terms of understanding a few things, like, dendrites are there to provide a bigger area for inputs. They're there to provide parallelization of processing of information to make location matter, as we discussed. So we have some understanding. I think there are still a lot of things that we don't know.
Yeah,
And
that's.
one of the things that I think is very important, and we know not that much, is the interaction of dendrites with glia cells, because I
Mhm.
bet there will be an influence on their computations. And glia cells are just starting to be studied, let's say, more extensively in terms of their processing and computations. And I expect in the years to come that we will know a lot more.
Hmm. Because there are also, I mean, now in like the standard sort of like caricature, well, function of a neuron is that only the only molecules, at least neurotransmitters and such, are released at the presynaptic synapses, right? But there are also molecules released from the dendrites, and it's all kinds of modulatory thing
Yep. Yeah.
also, which is also. But I guess that must be on a longer time scale, presumably, and maybe interactions with glia cells, and then you have something we haven't talked about is that the morphology, that you have spine dynamics, these little mushrooms, which where they
Right.
have the synapses, so they also vary in rather short time scales, right?
Yes, yes, yes. So whether a spine is a compartment is another open question. Let's say how much compartmentalization of current or chemicals or plasticity proteins do you have in the spine neck? That's also a big unknown. I
Mm,
guess we, with the voltage imaging techniques, we will be able to answer some of it in terms, at least, of the voltage spreading within nearby spines that could increase our understanding of, these
mm,
structures. And there is a lot of very nice work done in microRNA localization and production in spines by groups like Erin Schumann or Kristen Harris, for example. So, yeah.
mm. And then, I mean, we just had a podcast with Avrama Blackwell, and she's
Hmm.
done a lot of work on like subcell signaling pathways modeling, and so that's a whole like whole world in the inside cells, intracellular process in the neuron, and I mean they have typically there hasn't been too much connections between that kind of subcellular scale and the cellular scale when it comes to modeling.
That's true. And I think one big reason is computational complexity. You would need the compartmental models for that, so that you
Mm,
have, realistic, let's say, membrane properties and channels and receptors. And if you add to that models, which are already complex, what happens inside the cell, then, simulating them becomes a real problem.
mm, yeah, mainly for well the same kind of that you don't know parameters and you don't have computer time.
Yeah.
The same kind of limitations. One new thing which has come up, I guess, last decade or so is that we now have human neurons.
Hmm.
So what are they like? I mean, it's not only rodent neurons, but now we have human neurons.
Right.
What is it? Something magic element in the human neurons?
Well, I mean, judging from the behavior of humans compared to that of animal, there must be something fundamentally different. I
I
don't know
know.
if that comes down to the level of a single neuron or is an emergent property, of the cortex as a whole. What we've seen so far is that there are, I would say, not that impressive differences. I mean, okay, they are bigger, they are
Uh,
more compartmentalized, but they have similar ionic conductances. Maybe they have some difference in the membrane time constant. We thought that there was a unique type of a dendritic spike in neurons in
mm.
2020, but then it was also reported in rodents, so probably not.
So
Yeah. I
it's
haven't.
difficult. We are less and less unique the more we learn.
Yes. I mean, on one hand, this is good because it
Yeah.
means that you can still use, research in animals to understand the human brain, right? But on the other hand, it doesn't help a lot understand how consciousness emerges, for example.
No,
Yeah.
no. I don't know what kind of mechanism it should be at the neuron level that would help us,
Maybe
but
it's not at the neuron level. That's the
no,
thing.
it probably
Probably
not.
it's
Yeah.
not at the neuron level.
Yeah. Okay, we are approaching the end here, and I guess you, I mean, you have, you won't be out of work anytime soon. But what is
Hope
this
not.
experimental activity you're also starting?
So, I mean, we've wanted for years to be able to test some of our predictions experimentally. And although I have wonderful collaborators, I can
Mm,
understand that it's expensive and time consuming to go after what we care for. So,
yeah.
we decided that we will give it a try. And
Mm,
we've established an experimental branch of the lab that includes two photon in vivo imaging
mm.
in mice during behavior. So we went
Wow.
challenging
Wow.
for the challenging stuff, yes, and
Yeah.
we've been doing this for a few years, and we're studying flexible behavior in the frontal cortex of the mice. That's one project, and
Yeah.
another project it's the role of retrosplenial cortex in Alzheimer's. And in both cases we are looking obviously at how dendrites contribute
Wow.
to this
But how many
phenomena
people
behavior.
do you need to run a lab like? I mean, I don't know. It
We only
seems
have
like,
three people right now doing the
yeah.
experiments. Yeah.
Wow.
So it doesn't
That's
have to be
quite
very big. You really need smart people and
yeah.
highly motivated people,
Yeah. Yeah.
but not that many to
That you have.
run a lab, like this. So it's never going to be. We will not become experimentalists. It's just a small pet activity, let's say, of the lab to satisfy our own curiosity.
Mm, I see. Well, that's impressive. So it
Thank you.
seems like you still have some fire in your gut to
I
go for
mean,
that.
we have to
Yeah.
enjoy what we do, right?
No, that's true.
Yeah.
But I enjoy myself in other ways than trying to establish an experimental activity in our group.
Yeah,
So
yeah,
anyway,
can
yeah,
understand that. It's
yeah.
not completely trivial.
No.
Yeah, yeah.
So, but then you also have been making this, organizing this dendrites meeting for
Yes.
many years. That's
Yep.
good. Every year, somewhere
It's every
in
every 2 years.
every two
We
years.
had
Mhm.
the fifth event, so 10 years from its first meeting last May. It has been going on in Crete. In
Yeah.
fact, in this last edition, I kind of handed over a lot of the organization to a new set of organizers. This is typically funded through EMBO, and we have some rotation of organizers every few
Mm.
years. So it was led by Juliana Georgieva, Mark Harnett, and Lucy Palmer, and I was
Perfect.
helping them with the local organization. It's a great meeting. If you haven't been,
Yeah.
you should,
Yeah.
uh, especially if you're interested in dendrites. So I
Yeah.
think
I
it's,
am.
yeah, see?
Obviously. If not, I
So
wouldn't have invited you.
So it's
Yeah.
relatively small in the order of 150 people,
Mm.
but it's very focused on the latest developments in the dendrite field, modeling and experiments, and even
Mm.
some neuro-inspired AI.
Wow. Cool.
Yes, would
So
love to have you.
yeah. So well, thanks a lot for coming to the podcast, Jota.
Thank you, Gade. It was great fun to discuss with you.
Indeed, it was.
Let's hope people will enjoy it.
Yeah. I'm quite sure they will. At least I enjoyed it. That's why I make them.
That's what matters the most. Thank you.
Yeah.